In the brain, in all of our brains, there is a pathway which spreads from the midbrain here, nerve cells containing the transmitter dopamine, that project up to the forebrain to a region called the nucleus accumbens. This pathway is involved in making life worthwhile. Anything pleasurable is associated with activating this pathway. But it also turns out that a number of addictive drugs act by stimulating this pathway. And they do so to a much greater degree than normal physiological or social pleasures do. So that if an individual takes cocaine, there's massive release of dopamine here in the nucleus accumbens. And that similar thing happens with amphetamine, with heroin, with nicotine. The problem is that the nerve cell responds by downregulating its processes, weakening those synapses, so that the individual, when they stop using the addictive drug, are left with a suboptimal function of this mesolimbic pleasure pathway. So the individual actually feels unwell and suffers from what's called anhedonia, lack of pleasure. And the only way they can recover even normal levels of pleasure is to go on using the drug. So if we understand better how to regulate this pathway, how to block this downregulation or restore normal function, we would have a way of treating drug addiction that would be highly effective. The problem in drug addiction is not the individual actually stopping using the drug. Many addicts do that repeatedly. The problem is that they relapse, particularly in a situation which is familiar to using the particular drug. And one of the predictors of relapse is the amount of anhedonia that they suffer. Well, there's another side to plasticity that I mentioned at the outset, and that is stem cells in the adult brain and the discovery of neurotrophic factors which regulate them. I think everyone's heard of embryonic stem cells in the context of the ethical controversy about whether we should or should not use these cells or try to use these, to treat conditions such as diabetes or spinal cord injury. But there are stem cells in many other adult tissues committed to doing things such as making blood cells in the bone marrow, generating new cells in the lining of the intestinal tract, and so on. But quite a surprise 10 years ago to discover that there were neural stem cells in the adult brain. And this is a slide from the work of Fred Gage, who works at the Salk Institute in California. Incidentally, he's coming out next year to give the Kenneth Meyer lecture for the Howard Florey Institute, so you can hear him firsthand. But this diagram shows neural stem cells in two parts of the brain. It's hard to point out there, but here in a region called the subventricular zone in the forebrain, and these stem cells stream forward to a region of the brain called the olfactory bulb and generate new neurons. A similar thing can happen in the hippocampus, shown here, which, as I mentioned, is an important region for storing memory. A few years ago, two scientists here in Melbourne, one at the Walter and Eliza Hall Institute in Parkville, led by Dr. Rod Reitz, and the other part of the team was at the Howard Florey Institute, led by Professor Xiong-Sen Tan. They succeeded in isolating these stem cells, and this slide shows a nerve cell in red and a supporting cell in green, which had developed from one of these stem cells in culture. So now that we have these cells in culture and we can study them, we can try and find out what makes them divide, what makes them develop into nerve cells. If they develop into a nerve cell, what type of nerve cell? And so some of the dreams of being able to repair brain and other tissues, which until now has been science fiction, may really come to pass. There are a number of growth factors in the brain which regulate neural stem cells. The first one discovered was nerve growth factor by the Italian scientist Rita Levi Montalcini, and she won the Nobel Prize for that work. And a number of others have been discovered. One which I'll come back to is this brain-derived neurotrophic factor, BDNF, which has some very interesting functions which I'll mention in a minute. This shows some mouse ES cells in red, which have been coaxed by a particular combination of growth factors to convert into dopamine-containing nerve cells. Now, the same thing has been engineered with human ES cells, so that this would provide potentially a source of dopaminergic cells that could be used to treat Parkinson's disease. So this is very exciting. It's still in progress. It's still a long way from clinical usefulness, because we have to find out ways of suppressing those red cells in the background. If they were injected into the brain, they could develop into a tumour. So there's still a lot of technical work to be done, but in theory, the path forward of developing particular sorts of nerve cells to treat particular diseases is no longer science fiction. One exciting development in stem cells is another type of stem cell called induced pluripotent stem cells. This is quite exciting because you can take a normal cell from any one of us, could be a skin cell, culture it, add in 4 particular genes, and the skin cell will convert into an ES cell equivalent. And then you would have a ready source from the patient themselves for therapy. I think this is going to turn out to be a very big development. And finally, I'd like to tell you a little bit about Huntington's disease, or a mouse model of Huntington's disease. Some of you might have heard me speak about this before, but I'm doing so again because the implications of this particular experiment are so widespread for normal brain function and for other neurodegenerative diseases. So Huntington's disease is a progressive neurodegenerative disorder. It has motor effects, cognitive effects, the people become demented, and affective symptoms, they often become depressed. Its onset's in the 4th or 5th decade of life, and it's usually progressive and fatal. The genetic defect in Huntington's disease is known, and it's possible to take that gene abnormality and put it into a strain of mice. The mice then develop the same motor symptoms, the same memory loss as occurs in Huntington's, and even show depressive features. Dr. Tony Hannon at the FNI took some mice with this transgene and gave them an enriched environment, lots of toys to play with, things to explore, and compared the progress of the Huntington's disease in those with an enriched environment compared to those in routine housing in panels A and B. And what he found was quite extraordinary. This line is the acquisition of motor problems in the Huntington's mice with no enrichment, so that by 120 days, all of them were showing severe motor symptoms. The mice without the Huntington's disease, labelled WT, performed perfectly on this task up to 160 days. what was extraordinary was the animals with the mutation and an enriched environment showed a remarkable delay in the onset of the symptoms, and when they occurred, the symptoms were less severe. And this was accompanied by a delay in actually developing the disease. And Hannon believes that what's happening in Huntington's disease is that the neural stem cells are impaired in their response due to lack of a neurotrophin BDNF, and that environmental enrichment partially rescues this condition and certainly could make a significant difference to people with the disorder. And one final example of growth factors is in depression. And very surprisingly, BDNF is turning out to probably have a role in depression. In depression, there is reduced neurogenesis, there is lower levels of BDNF. BDNF itself is antidepressant. And very interestingly, exercise, physical exercise, is one of the many ways to boost BDNF. And we know that exercise is antidepressant. It's very likely that mental activity as well has beneficial effects on the brain through the same mechanisms. So there have been dramatic advances in understanding the brain at the cellular and molecular level in the last 10 years. We know a lot more about the chemical basis of neurotransmission. We're learning that the brain's connections are plastic. They're highly variable depending on how they are used. Neural stem cells have been discovered in the brain, and factors which regulate them are being worked out. The structure and function of the conscious human brain can now be probed using techniques such as functional magnetic resonance imaging. The brain's much more plastic than we previously believed. Initially in life, our genes that we inherit from our parents are involved in laying down general pathways in the brain. And during development, there's an abundance of synaptic connections develop. These are pruned by use-dependent mechanisms so that we all end up with the brain that is appropriate to the environment that we found ourselves as a child and find ourselves as an adult. And similar processes are involved in recovery of function after neurodegenerative disease or injury. Exercise, both physical exercise and mental exercise, have beneficial effects on the brain. They affect the way it develops. Exercise is antidepressant, as I mentioned, and stimulates neurogenesis. Repair mechanisms dependent on neurogenesis are stimulated. And in case of Huntington's disease, we've got good evidence that environmental enrichment slows neurodegeneration, and we believe the same thing is true in humans with Huntington's disease, as well as almost certainly those with Alzheimer's disease. So contrary to the dogma that the brain can't repair itself, it's a highly dynamic and plastic organ, and you have a lot of control by the way you live, by the way you use your brain, on the sort of brain you end up with. Understanding the molecular mechanisms of neurogenesis and synaptic plasticity brings the promise of greatly enhancing repair and injury after injury or degeneration. So I'd like to stop at that point and answer any questions you might have.
Is there any proof or research done on more unconscious mechanisms within the brain being able to recover functioning. So my understanding of neuroplasticity is that it's use it or lose it. But what about unconscious functionings like breathing? If there's been difficulty or damage there, is there any research?
Any work on that?
Yeah.
No, it's a very good question, because a huge amount of what our brains are doing we are not conscious of. Even vision, sensation and so on are conducted largely at an unconscious level, and a small amount of this gravitates up. The sort of unconscious processes you were talking about, such as breathing, life support activities, are probably things that you've either got functioning or you don't survive. But in another sense, the higher functions, unconscious functions, are probably what's being changed in psychotherapy and psychoanalysis. So these techniques, when they do work, rely on plasticity about largely unconscious processes.
I have a rather left field question. Would you like to comment on where lateral thinking comes in the operation of the brain and the whole de Bono philosophy?
Lateral thinking is when a connection or an association pops into your brain that isn't apparently a linear, rational step. And the thing about our brains is it's doing it all the time. Our brains are hypothesis generators, if you like. The idea that we're a rational machine that calculates what's the best action and takes it is a complete myth. Most of our brain activity is in making lateral connections, and this is what makes us so unique and so much better than a computer at solving problems. And I think if you looked at what's going on in the brain, most of it is lateral associations.
My question relates to the implications for education and the education system. Does your research indicate that there would be significant implications for how children as well as adults learn?
Yes, I think there is. When you look at children, they're absolutely hungry. Well, adults are hungry for knowledge, and hence you've come out on this dark, wintry night. But in childhood, this desire to know and understand is even more intense. I think we've got education completely the wrong way around. We take children, put them in a room and tell them what we want them to know at that time, they're in some other space, curious about this, gazing out the window. If we were able to build an education system in which the teacher responded to the child's questions at appropriate stages and then followed on, I think we would breed a race of geniuses. The problem is that that's not a very practical thing to do, but it's interesting that a number of extraordinary thinkers have been educated by their parents, suggesting that sort of process happened. Yes, so I think that's right. We're losing great opportunities in early development to boost brain function.
I just have a question about early childhood, because part of the revolution in looking and the re-emergence of the importance of early childhood has been based on the new brain research. And yet, ironically, it sort of perpetuates early determination and doesn't really recognise the plasticity that you've talked about. And I wonder why we revert to this sort of notion of, the early determination as opposed to what really should be the view, which is a life course view of human possibility.
Yes, I don't disagree with that at all. I think I emphasise the early childhood plasticity because it's very dramatic, but as you say, this plasticity continues right throughout life, and anyone in this room is capable of learning a new skill or honing an old one. And there's a lot of evidence that doing so is actually protective to your brain. Thank you for that comment.
Professor, most of the examples you've used have been about clinical and pathological interventions. Is anyone applying this research into normal decision-making? Say business decision-making, for example?
I'm not aware of that, and I don't think there's a lot going on, because I'm aware of an approach that's been made by someone with the interests you've just mentioned, to work with a neuroimager to begin to look at how the brain changes. Of course, companies are also interested in how the brain responds to advertisements, so they can make better ads. So there's all sorts of implications of this new technology.
So in stroke recovery, do you ever see, in regards to plasticity, the changing side, say, with language of the brain? Do you see language going to the right at all?
Yes, there's some very dramatic work by Professor Graham Jackson at the Brain Research Institute out at the Austin Health, where he studied people who have undergoing elective surgery to remove a lesion, an epilepsy-causing lesion, right near their speech area, Broca's area on the left hemisphere. And in that person, he saw quite remarkable shifts in speech activation around the time of surgery and on follow-up 12 months later. This plasticity of maps, if you like, is much more general than just the motor example I showed you.
I wondered if you could comment on sometimes the ability to reach people with Alzheimer's. I had a friend whose husband had his finger jammed most severely in a door and was totally lucid for 24 hours, told the staff the story of his life. I mean, you can't go around hammering people's fingers, or even the occasional question that elicits an immediate response. I mean, all the information is still there.
Yes.
What goes on?
It's fascinating that in dementia, whether it's due to Alzheimer's disease or multiple small strokes or many other causes, that recent memories suffer severely. So the person may not remember you visiting them yesterday or even an hour before, but they can still remember their daughter's fourth birthday, and the distant memories are very much preserved. But in terms of injury, you bring up an interesting question. It's known that patients with Alzheimer's disease request less pain-killing medication than other people. And a study done at the FNI by Gary Egan's group looked at brain activation in response to a mildly painful finger stimulus. And they found that in people with Alzheimer's disease, those pain pathways were fully intact, including the regions of the brain that we know are related to the sensation, the unpleasant sensation of pain. So it seems that these people are complaining less, but they're probably suffering more. So the message is nursing and caring staff need to be aware.
Professor, I'm just wondering, in the case of someone who has suffered an acquired brain injury, who perhaps has lost the ability to concentrate and may have their motivation and drive has been impaired, how do they actually get to the next phase of doing the brain exercises where they're grappling with the ability to concentrate and, be motivated enough to actually go ahead to do these programs? I was just wondering how that person overcomes that initial phase.
Yes, it's quite a difficult area that you brought up, but occupational therapy, when I was a student, we used to disparagingly call it basket weaving and saw it as a soft science. But in fact, it's looking at those very processes. What's the most effective way to engage these people in activities? And I've visited people after acquired brain injury and found this person had motor deficits, cognitive deficits, coordination problems. And he was standing at a table doing this task. And I realised that the occupational therapist was simultaneously training his standing, his balance, his coordination, and his cognitive abilities. So these are areas that are really being taken seriously now.
I'm interested in your comments around early literacy earlier, but also around some of the research that's been conducted that early reading experiences help the brain to learn. So we know that, children that have rich literary environments when they're young obviously do better academically. But I'm wondering about the learning to learn part and the role that reading plays in that, particularly for young children in that sort of 2-year-old phase that you were showing us earlier.
You're absolutely right. I think reading to young children is an enormously potent way of capturing their interest in learning and in books. And what better place to say that than here? And the interesting thing about brain plasticity and learning is that skills acquired in one area, such as reading, seem to translate to increased efficiency in other areas. So it probably doesn't matter too much what sort of activity you're involved in and learning, but the mere fact that you are.
I was wondering what you attribute the rise in neurodegenerative conditions to.
Well, particularly Alzheimer's disease, there are a number of things. I think we recognise it much more than we used to. When many of us were children, it was believed that as you aged, you lost your marbles and became forgetful. Now we know that there is no decrease in cognition with age. It's only if you develop a disease such as Alzheimer's disease or other dementia. But that aside, there is an increased incidence in Alzheimer's in the community because it goes up with age. And many more people are living into their 80s, having survived infectious diseases or cardiovascular diseases, to become prey to neurodegeneration. So this is an extremely important area to research and to work on, to make progress in. In that regard, here in Melbourne, at the Mental Health Research Institute under Professor Colin Masters, is some of the best work in the world going on in Alzheimer's disease. And because of the deep understanding of the biochemical processes involved in Alzheimer's disease, I think there's a realistic prospect of a cure, meaning a medication that could be taken to stop progress of the disease, even within the next decade.
In the event of an accident and the damage of the brain and it broke off the fibers to your sense and smell, is there any way of regeneration on that?
No, the question was, after an accident, losing the sense of smell or anosmia. This is a common injury where the frontal lobe moves across the olfactory plate and shears off these nerve connections, as you say. I'm not aware of any treatment of that, but it's certainly one of the things that may well be treatable in the future using neurotrophins.
Will you please join me again in expressing our thanks to Professor Mendelsohn.
Thank you very much.